Hello friends, welcome to my channel Clinical Biochemistry by Dr. P.K. Prabhakar. Today I am going to talk about one of the another variant of PCR, and that is nested PCR. In the last lecture we have talked about one of the variant of PCR, that was multiplex PCR. So this is the basic simple PCR, normal conventional PCR which we can call it, which have three different stages. We are having one of the stages, denaturation stage, where a double-stranded DNA will be denatured and it will give you single-stranded DNA, which will work as a template. In the second stage we are having annealing, where temperature will be slightly lower than the denaturation step. In case of denaturation, temperature will be 94 to 95 degree. In the case of annealing, where primer is going to bind with the single-stranded DNA which we have received from the first previous process, there primer will bind with the template DNA. And in the last stage, extension will be there, where thermostable DNA polymerase is going to extend the primer by using single-stranded DNA as a template. So this is the same thing. We are having three steps. This is the first stage, this is the second step, and this is the third step. In the first step we are going to denature, so this DNA will be denatured into two single-stranded DNA, and then we are going to have reaction. In the reaction mixture we will have primers, which is specific for both the strands. So for this strand also and for this strand also we are going to have template. Then thermostable DNA polymerase, we may have different types of thermostable polymerase. Normally, most commonly, most preferably we are using TAC DNA polymerase, which is coming from the Thermus aquaticus. But other than TAC polymerase, we have some number of other DNA polymerase also which can be used in the PCR reactions, like it will be Vent polymerase, PFU polymerase, PWO polymerase. And by using these deoxyribonucleotides in the presence of this enzyme, TAC thermostable DNA polymerase, this first primer will bind there, and then this DNA polymerase is going to extend the primer by using these nucleotides in the specific reaction mixture, or you can call it environment, microenvironment you can call it, where pH will be maintained and in the presence of divalent metal ions. So ultimately from this strands we have got two double-stranded DNA, and this will go for next cycle. Again, this next cycle means it will go for denaturation, it will go for annealing, it will go for elongation. So this process is going. This is the conventional PCR. What we are talking about, the nested PCR. Nested PCR is normally it is basically simple PCR, but there are certain modifications. So the specificity is the main aim when we are talking about the PCR, because you are going to elongate the DNA template. Whatever template you will have, that template will be produced in the very high quantity, very high number. So if you don't have a specificity, then undesired DNA will be amplified. So that's why specificity is one of the main focus of the PCR reaction. And whenever we are going to modify a PCR reaction, conventional PCR reactions, means we are going to increase their specificity as well as the sensitivity of the reaction. Specificity means only the specific target DNA will be amplified, and sensitivity means it will be very precise reactions. Nested PCR, it is a simple and easy modification of the conventional PCR, which actually increases the specificity of the reaction. Means only the target DNA will be amplified. So to do this specificity, to increase this specificity, we are having nested PCR. Actually, what is the nested PCR? Here we are going to increasing the specificity of the PCR reactions by reducing non-specific binding of the primers. So normally whenever we are having a template DNA, so primer, this is my target DNA. So actually we focusing that our primer will bind here in the target DNA, within the target DNA. But it is not necessary that every time this primer will bind here only. This primer may bind here or may bind here. So this is called a non-specific binding. So to avoid this non-specific binding of the primer and their elongation at the end of the PCR reactions, we are having this nested PCR. So in this nested PCR, we are going to increase the specificity of the PCR reactions by decreasing the non-specific binding of the primer. And how we are going to do that one? So there we are going to use two specific primers, means two sets of primer. In conventional PCR we are going to use only one set of one pair of primers, but in the nested PCR we are going to use two sets of PCR. So the nested PCR is the modification of PCR intended to reduce non-specific binding in product due to the amplification of unexpected primer binding sites. So the nested PCR is the best choice for microbial identification. Whenever you want to identify microbes and 16S RNA analysis, that time this nested PCR is one of the best choice. Now in this nested PCR we are going to use two sets of primer. So this two sets of primer is going to give us very high sensitivity in this reaction, and here both the primer have different functions, different properties or unique properties you can call it. The first set of primer, which is called as outer primer set, they binds outside the target DNA. Like if this is my DNA and this is my target DNA, so the first set of primer will bind somewhere outside this target DNA, and that's why they are amplify a larger fragment of DNA, and that's why it is called as outer primer. This another set of primer, which is the second set of primer you can call it, they binds very specifically at the target site, so that will bind in within the target range, and this go for this range of reaction. The first outer primer binds to the target DNA, means in our template DNA outside the target DNA, and amplifies it, and after that one, whatever product we are getting after the first cycle of amplification by using this outer primer, that DNA we are going to use this second set of primers which binds within the target and amplifies it. So this second set of primer is called as inner primer. So first outer primer will be used for the amplification reactions, and then this inner primer will be used for amplification. Here in this case of nested PCR, our template DNA is the primary binding site for outer set of primer. So whatever template DNA, whatever target DNA you are going to use, so this DNA, complete DNA will be used as a template target DNA for the outer primer, and whatever product you are getting after the first cycle by using this outer primer, that we are going to call amplicon after the amplification process. That amplicon will be the target for the second set of primer. So the for first outer primer, our complete DNA is the target. For this inner primer, only the amplicon of the first reaction will be the target. We will see that one. So this is the basic simplification diagrammatic representation of the nested PCR, where this is my target DNA. So first round, first DNA, first set of primer, which is called as outer primer, it will bind randomly at outside the target DNA and do the amplification reactions. In the second set of once whatever product we are going to get by using this outer primer, there we are going to use inner primer, and inner primer will bind within this template target DNA and template target region and then amplify it. So that specification we are getting it. So this is the slightly. So this is one strand of DNA. This is second strand of DNA. This shaded region, this dashed square rectangle region is my target DNA. So the first primer, which we are going to call outer primer, they are binding somewhere outside the target DNA and amplifies it. Whatever product you are getting in the first cycle, so means PCR you are going to run two times. First time you are going to run with this use of outer primer, and then we are going to use inner primer, which normally binds within the target DNA and amplifies it. So now we will see some of the specific feature in this one. In this case, in the case of nested feature, this techniques requires only those things which we require during the conventional PCR, nothing extra. So it does not require any additional reagent, any additional chemical or additional instrument. It does not require than the conventional PCR. Only what we require extra is one extra pair of primer, one extra set of primer. That is the only additional things we needed in this case. And in the conventional PCR, normally we may face some of the issue, like the common problem in the single set of primer or the conventional PCR is the early activation of Taq DNA polymerase, primer dimer bind formations, or non-specific binding of primer. So to avoid all these things, we can go for nested PCR. So in the nested PCR, we can remove all these disadvantages which normally we face in the conventional PCR. So we will see both stepwise process. The first, outer set of primer. The outer set of primers are that are upstream to the inner set of primers. If you will see, this is the target DNA and this is the inner primer. So normally this outer set of primer binds upstream to the inner set of primer. This is the inner set, and upstream to this one is we are having the this outer primer binds. Then outer primer binds to the outside of the flanking region of the target DNA, and the first round of PCR, it is possible that primer can bind somewhere outside also. Means not only whatever we are seeing here, only 2 places, but it can bind at 4, 5 places, 6 places, 7 places, but many number of places, and normally amplifies it. So here you can get many DNA bands may be observed, and those things are normally false positive because we don't want all those things. So if this is my target DNA, template DNA which we have used, so and this is my target DNA, means which we want in the result. So when we are going to use this outer set of primer, it may bind here also, it may bind here also, it may bind here also, it may bind here also. So many. So in the case you will get amplification of up to this one. In case you can get the amplification of this one. So many different small DNA bands you can see during the gel electrophoresis, and those are because of the false, they will give you false positive result. So this is that outer set of primer and how we are going to use it. Then we are having second set of primer, that is called as inner set of primers. So even if the non-specific DNA is going to be elongated, means whatever in the first set we have got some kind of false positive result because of the non-specific amplifications, so that will be removed here in the case of when we are going to use inner set of primer, because this inner set only binds with the target DNA. So non-specific DNA will not be amplified in this case, because in the previous case whatever extra DNA you have got it, there this inner set of primer will not bind. So this will bind only with the target DNA, and only target DNA will be amplified. So the second set of primer is specific to the inner sequence, that is the amplicon for the first round of PCR, and it is very unlikely, very less chances are there, very almost negligible chances are there that the inner set of primer binds to the other than the specific site, other than the target site. The amplicon from the first round of PCR is the template for the second round of application. So whatever product we are getting the first PCR by using outer primer, that will be used as a template here in the case of second round of PCR by using inner set of PCR. So this is the normal diagrammatic representation. This is our DNA, where this is the outer forward primer, this is the outer you can call it reverse primer. So it binds at many places. Now, so in this case it is going to be amplified different. This is non-specific amplification, this is non-specific amplification, and this is also amplification. So we have got 3 different kind of DNA product. So this is one product, this is second product, this is third product, but this is after the first round of PCR. But when we are going to use second round of PCR by using this inner primer, inner set of primers, that will not bind in this case, that will only bind within the target region. So in that case, in the second round of PCR, only this fragment of DNA will be amplified. So this increases, this second round of PCR is going to give you the specific desired target DNA. So this increases by using 2 round of PCR, or by using 2 set of primer and running 2 set of PCR, we are going to increase the efficiency, increase the specificity of the amplified DNA. Now what is the protocol? So what we do, this protocol, this nested PCR, as you know, the normal PCR, conventional PCR has been discovered by Carey Morris in 1985. In the 1993, Pommel Varin and his co-worker described this method by using 2 sets of primer for increasing sensitivity and both the specificity of the PCR. Here we are going to use 2 steps. So this reaction will be completed into 2 steps. In the first step we are going to use outer forward and outer reverse primer. After that one we are going to use this outer set of primers, and then we are going to run the PCR, and we are going to get some products. So after the completion of the first round of amplification, first round of PCR reactions, take the tube and prepare the reaction for the second round of amplification. So whatever product will be the from we are getting from the first round of PCR, that will be the target or template for the second round of PCR. Then in the second round of PCR we are having two options. Either we are going to add directly 1 microliter of inner forward and inner reverse primer into the PCR reaction tubes itself, whatever we have got, the pen drop. In the same tube we will add this much primer, or from this tube, pen drop tube, we will take some sample and run a fresh set of PCR. So 3 ml of PCR product will be taken from the first amplification and use it as a template in the fresh pen drop. So both way we can do it. For both the first reaction and second reaction, normal PC, this is the normal PCR processes. So same process we are going to use. First initial denaturation, then denaturation, annealing extraction. This is called as one cycle of the reaction. So this cycle we are going to repeat it multiple times. Normally, when we are running the first set of primer or outer set of primer, that time we are going to run 25 cycles, and for final extension or the desired extension, or the you can call it a specific extension of the target DNA, there we are going to run little bit more cycle. So the second time when we are going to run second cycle, that there we are going to run more than around 35 cycle of the PCR. What is the advantage of this nested PCR? So it is mostly beneficial in the studying of the polygenetic analysis or genetic polymorphic analysis. The main advantage of this method is it gives you 100% accuracy, specificity and sensitivity. So this is the most specific advantages of these methods. But for the impossible template where GC content is very high or the chances of non-specific binding is very high, nested PCR offers the best result. Then it is also useful for the amplification of DNA with the low abundance. Further, the nested PCR is the best choice for carcinoma and viral infection studies. But along with these advantages, there are certain disadvantages also there. So this method is very time consuming. It requires more extra reagents, so it is a costlier process. You can call it not cost effective, it is costlier process, where we are going to use one extra set of primer, we have to run gel electrophoresis, we have to use nucleotides, we have to use many things. So it is requiring reagent needed, means the need of reagent will be more, so it is a costlier process. And last is the chances of contamination is also higher, because pipetting you are doing several times. So more opening, more handling, so more chances of contamination will be there. So these are the disadvantages of this nested PCR. Then how this nested PCR is going to be used for the microbial identification. So the first, we are going to use nested qPCR or the nested RT-PCR, because we are going to use their 16S ribosomal RNA or 18S ribosomal RNA in case of eukaryotic identifications. So that's why we require RT-PCR. So it gives a great power to this technique, which can be helpful method for the phylogenetic analysis and identification of the different pathogens. This technique has high sensitivity, and hence even if the sample contains very low DNA, it can amplify, which is not possible in the conventional PCR. This in addition to this method is very high specific as well as sensitivity. In this nested real-time PCR, the universal primer we are going to use, that is for against 16S ribosomal RNA or 18S ribosomal RNA, and there for this one or for this one we are going to use as a outer primer. So here this is the pictorial things. So if we are going to have 4 different species and we want to identify them, so this is my first set of reactions. So where we are having 4 different DNA, 4 different ribosomal RNA we have selected, and that will be amplified by using a target, means you can call it outer primers. So this is universal primer, outer primer has been used. Then once this whatever product we have got after this reactions, so once it is amplified into the PCR machine, the set of specific or unique sequence primer used as a inner primer set. So in the first cycle we are using a common, means you can call it non-specific or universal primer, which will work as a outer primer set. And whatever product we are getting after this first cycle, that there we are going to use inner set of primers An inner set of primer, what is the inner set of primer? That is either a species specific or that will be unique unique means very a specific sequence, and that will be used as an inner set of primer. The second round of PCR or the multiplex PCR. Multiplex PCR already I have discussed in my previous video. So the second round of PCR where we are going to use this inner set of primers, individual PCR or the target specific PCR can be applied for this one. The unique sequence primer are specific to one pathogen, because when we are using a specific, so that will increase only one DNA, which amplify the template DNA if the target sequence is present, and if it is not present, it will not be amplified. Once the amplification is achieved, the amount of pathogen present in the sample is measured quantitatively, and ultimately the species of pathogen can be identified. So this is how it is very useful in phylogenetic analysis or identification of the microbes or any kind of pathogens. So this is the picture. Whatever product we have got from the first cycle, so that will be so this is from the first set of PCR where we have used a universal primer as a outer primer, and there we are going to use a second PCR cycle where a specific primer we are going to use, and there only target DNA will be amplified according to if it is present there. If it is not present, target DNA is not present there, it will not be amplified, and that will be detected by the gel electrophoresis. So this is all about the nested PCR. Hope you have understand it. If you have any query, any comments, anything you want to share, you can write in the comment box. If you are new to the channel, you can subscribe it. If you like the video, press the like button, and you can press the bell button so that you will get all the notification on time. Thank you very much. Have a nice day.